preclinical models to clinical doses. And before that, I'll introduce myself. I'm Sri Kidambi. I'm an Center Scientific Director. And
Deidre Dolmas, a distinguished fellow at Boeing Ring.
So what we're really talking about was how to connect, and that was a discussion. We had an open question: is dose translation fails not because of we lack models, but we lack standardization, qualified, and context-defined systems. So the question was really addressed was if you want to have clinical confidence, regulatory confidence, we need certain aspects of how does that need, and those were the questions, 3 questions, which is very similar to what the discussion you had. The first was really looking at unmet regulatory needs and the questions that need, of interest of statements to really drive that part. So we're not really discussing about the models in our breakout session. The intent was really the dose translation from these preclinical models, exquisite models, which is not a lack of biological ingenuity, but now how does it change into decision-making tools? So some of the things discussion came out was what context of use should complex in vitro models, CIVM, support in clinical dose selection, and that ranges from first in human doses or dose escalation limits, toxicity thresholds. So, and that's really going to be something of question of interest and also driving the decision-making capability of these tools. What validation evidence would regulators require to trust dose predictions generated from these NAM-based models, such as organoids or tissue shapes or any other model, even digital twin? What performance benchmarks define acceptable predictive accuracy for translating preclinical exposure relationships to humans? So really driving the point of extrapolation or interpolation. How do you connect the dots from what we see in humans and how do we see that same response in these in vitro models? And how should outputs from complex in vitro model systems integrated with PBPK and PKPD models, because that's going to be critical. So it's going to be mathematical models which has been put together. So how does we use those? But how does it connect the quantitative dose prediction? And that was some of the unmet regulatory needs, and what standardized characteristic metrics are required for organoid or tissue systems, something we talked about, but again, in silico and other type of NAMs to ensure cross laboratory metric comparability, especially from FDA metrics. And next slide.
Yep, sure. All right, so our next was that we're looking at the various different opportunities that we have across the various different systems. So some of the opportunities that were discussed were really looking at these multi-site benchmarking and through these various different, say, consortia efforts, and how do we then bring these, different efforts together to help better benchmark, where each individual, consortia is working on something, but can they then collaborate to have these larger data sets? And these larger data sets will be able to help us establish more, I would say, ranges or valid, I want to say validated, but more, I would say wider ranges where we have more confidence in, say, potential biomarker levels that we would expect to see when you're translating between in vivo versus the in vitro systems. I'm looking at integrating the animal and non-animal data to clearly define the targets that we're looking at. How do we then incorporate those models and use something similar to, say, QSP modeling that is getting done for determining clinical doses? We have opportunities to be able to do that and incorporate that data from the various different models that we're generating. So that is currently, an opportunity that is out there, that some there's, more models can be developed in that way. We're generating, AI, other ways to do that. And then how do we incorporate that human, aspect into that too, to make sure that we're doing that the proper way. Looking at different harmonization of definition, so similar to in the past, where if you're looking at images, if you're looking across these models to qualify these models or more so characterize them, similar to pathology in the past, where pathologists had different terms for things, looking and saying, okay, well, how do we then harmonize that a little bit more so that you can compare when you read something in literature, what output that is, and then how do you correlate that to the various different efforts. And then looking at, and then again, some opportunities in the biological parameters and looking into PBPK and how do we evaluate these endpoints within these models, there's a lot of opportunities to be able to do that, to better help characterize them, and then correlate the effects that you see to help potentially establish those more translational clinical doses. I think that was everything on there.
And the gaps of challenge was basically very mirroring what we discussed at opportunity. So the same thing came recurring. So the same issue is both the opportunity as well as the gaps and challenges for in terms of preclinical models to dose, clinical dose translation. The challenge really is, again, fit for purpose or context of use. Those are mean different, but at the same time, how does that biological fidelity in current systems for critical dose translation functions? And again, the question ranges from metabolism, looking at metabolism or transporters or immune responses. So what are those criteria? The variability across platforms and laboratories and protocols that undermines reproducibility, that's really a challenge. And from the confidence decision-making capability, that is really a limiting factor for FDA and other regulatory and also clinicians to adopt these preclinical models, especially in vitro models. Lack of standardized reference compound panels for benchmarking predictive performance, and the rest of the two.
Where are you at? Yeah, so then also looking at the integration of the outputs into, I would say, these pharmacokinetic, pharmacometric type models. There's opportunities to do that where you can integrate both clinical data, in vivo preclinical data, as well as the in vitro data. And that's done, that is starting to get done. Just say, if there is no pharmacologically relevant model, there's a lot of in vitro systems generate some sort of model that will then recapitulate and be able to predict. So integrating all that data together, and then also looking at these various different types of data sets that are inconsistent, that potentially don't have similar metadata. And that's a big gap, I would say, across the various different areas, and a challenge. So how do we better be able to I would say characterize different endpoints and be able to help with model validation, because doing so and having say similar terms or consistent terms will be able to help us along the way across various different types of models. Even if you're looking at liver versus cardiovascular, understanding the types of standard terms that get used for those, I think would be go a long way in something that was discussed.
Really, to finish up more from a perspective of shellfish SOM related stuff, it would be appreciated if any data, because that's really what we were talking about, data, and what metadata is incomplete data. That's a challenge. We are really driving the AI machine learning as a data optimization process to generate protocol optimization. And from an organoid perspective, if there's any data available and really from a perspective of benchmarking, the clinical data, that'll be really great for us to really push this key, but really drive organoids. And again, this is really be a test run, if you will, pilot study for organoids, but then use it for other in vitro model systems too, to push from artisan technology, which is organoid which is amazing biological, nothing against the amazing biological ingenuity infrastructure available, but now move it to a decision-making tool. So that's really would be good. So if you have any data, want to talk to us, Jack is right here. He's AI machine learning director and the SOM director, so please reach out. I'll be more than happy to talk and get your data. Thank you.